Okay, so now that we finished the cell shape, we're going to move on to their external structures. So one of the key external structures is any sort of appendage on the outside. So appendages are accessory structures that will sprout from the surface of the bacteria. They are typically designed to assist in mobility and motion, or the bacteria's ability to stick to or adhere to a surface. Mobility is just the bacteria's ability to be self-propelled. So as you can imagine, it would be important that they be able to go where there's nutrients, where the pH is correct, where the temperature is correct, and not just be at the whim of their environment. One of the key tools they have for mobility is a flagella. And I have it here that they are basically the bacterial propellers. So if you think of like a little prop plane, basically having a single propeller, same kind of thing with the flagella. And the flagella is comprised of 3 distinct parts. It has a filament that's kind of the whip-like helical structure of proteins that we actually think of as the flagella. It has a hook or a sheath. This is kind of the like L-shaped bracket you'll see in a minute, and the basal body, which is actually where it hooks in to the cell membrane and the cell wall for anchoring. Okay, so if we zoom in here a little bit, we have the filament. That's the helical set of proteins that's actually going to be performing the whip-like motion. We have the hook out here, this kind of L-shaped structure that's going to be the rotating component. And then all of this is inserted into the membrane and the cell walls here with the basal body that is comprised of a rod with several rings. All spirilla or spiral-shaped bacteria will have a flagella. About half of the rod-shaped bacillus ones will have them. And then there's a couple of coccus bacteria that have flagella, though it's pretty rare. They've also been found in a variety of locations. So some will have only one flagella on one end, some will have them on both ends, some will be all over the place like a bad hair day. Okay, so there can be more than one or only one, and they can be in a variety of locations. If they're found on either end, we will refer to the flagella as being polar. Okay, so here's some of the examples of the arrangements. We have monotricious. This would be a bacteria with a single flagella. So like this picture in A here, we have a single protrusion out here as its propellant. A lophotricious bacteria would have a small little bunch of flagella on one end. Okay, so think, I don't know, like a little minion, okay, with like out here as its little hair on one side, okay, and then we have the amphitricious where there's flagella on both ends, so it'd be polar, and we have peritricious where they're distributed randomly all over the entire surface of the cell. Now, it's important to note that flagella are very difficult to visualize with light microscopy. So last time we talked about how do we look at cells on the microscope, and staining the actual cell is pretty straightforward, but getting these individual little protein protrusions that are very thin is very difficult. So you'll notice that a lot of these are taken with scanning or transmission electron microscopy, not with light microscopy. So if you did get the light microscope to work, that's great. But one of the ways that we test for mobility that's pretty quick and easy is with semi-solid media. So you would make agar in a tube and you would make it not super solid, not like a jello, and you wouldn't make it really liquidy. You kind of make it in between, and you take a needle and put bacteria on the needle, stab the media, pull it out, and then let it incubate. And then when you came back, you could see, did the bacteria grow through the whole sample? That would tell us it has a flagella, it's mobile because it moved from where I stuck it. Or if it only grew in the little stab mark, then we know that it's not mobile and it could not propagate through the media. Okay, so one of the big goals of having the flagella is to respond to the bacteria's environment. And chemotaxis is the movement in response to a chemical signal. So if the bacteria senses, hey, there's more food in one direction, that would be a positive chemotaxis, when it would start to move the cell in the direction of a favorable chemical stimulus. Like, hey, there's glucose over there. Woohoo, we need some of that to make some ATP. And it would move towards that chemical signal. We also have negative chemotaxis. So maybe instead it senses that there's an antibiotic, or maybe we've sprayed the surface with Clorox, things like that. It would be moving away from a potentially harmful compound. Now, bacteria take a very unique approach to their mobility. So it's not like they're just chugging along in a very straightforward fashion. They actually perform this run and tumble like motion. Okay, so to understand this, you start first with your little bacillus bacteria here with its monotricious individual flagella, and it's moving along towards a chemical signal, a positive chemotaxis. And then maybe it senses that, oh, wait a second, it's more down here, or maybe I didn't read the chemical signal right. It will get kind of confused and the flagella won't move in a defined direction. It'll start moving the other direction. It'll be a little confused. It will tumble, and then it will right itself and begin moving again in another direction. This is also true with a peritricious mobility. So if it has multiple flagella all over the surface, it can again be moving in a defined direction. And then if the flagella get a little confused because it's getting mixed signals, then it can tumble a little bit and turn itself around. Okay, so this is what it would look like if we were tracking its mobility. So you'd have, I'm confused, I'm tumbling, running in a very defined direction, and then getting a mixed signal again and tumbling. And so you can see how it moves in a linear fashion, stops to think about it, moves again, moves again, moves again. So it's not a very fluid sort of mobility. Okay, so appendages can also be used for attachment in addition to helping with mobility. There's three main ones here that we've briefly discussed. The pilus, which is a long tubular structure produced by bacteria for conjugation. So this would be kind of that little straw-like structure we saw earlier that can be produced if a bacteria wants to share a plasmid with its neighbor. Fimbriae are short surface appendages that are used for adhesion. So we see them over here in yellow. They help the cells stick and not get washed away or wiped off a surface. And nanotubes, these are small channels that are made in the bacterial membrane that can help it share nutrients or ions or energy, ATP, with a neighboring cell. Okay, so to zoom in a little bit more on the pilus that is used for conjugation. Conjugation is when bacteria contact with their neighbor to share genetic material. So we would have a donor, like let's say this one's the donor, and it would make the pilus and extend the protrusion out to its neighbor, and then it could send the plasmid, that circular piece of DNA, through the pilus to the recipient cell. You'll also notice in this picture, this kind of haze on the outside. This is the little hair-like fimbriae protrusions that are helping the bacteria stick to the surface. This idea will come back again. So be forewarned, figure out what conjugation is now, because it's going to come back multiple times throughout the semester. Another surface coating to be aware of is the S layer. This is a layer of many, many copies of proteins linked together on the surface of a bacterial cell. So you'll see it here kind of in this gray. It kind of acts as like a chain mail to protect the bacterial cell from a hostile environment. You'll also see the glycocalyx out here in green, which we're going to discuss soon. The glycocalyx develops as a coating of repeating polysaccharides or glycoprotein units. So it's going to have a combination of glyco being like a sugar molecule and proteins or amino acids together. It's very defined and orderly, but it's very complicated with lots of repeating units. This layer gives protection and helps it to adhere to the surface. And there's varying types of glycocalyx layers. You can have a slime layer, and this would be the diffuse, kind of disorganized layer that helps bacteria, basically just keeps them from drying out. So if they're out on a surface, they're going to keep them moist. If it's more defined, it's more tight and dense and orchestrated, it would be known as a capsule. You can see the capsule out here. Again, the capsule assists a lot in keeping the cell from being phagocytosed or eaten by our immune system. Okay, so capsules are often formed by pathogenic bacteria, and bacteria that have a capsule have a greater chance of causing disease, again, as I was saying, because they can evade the immune system and avoid being phagocytosed by white blood cells. Capsules also help bacteria form biofilms. Okay, so we have a little picture here on how a biofilm forms. You can see a surface, maybe it's something, I don't know, like a catheter or a table in the hospital, and you have your surface here. And then you're going to have your first colonists. So the first couple of bacteria that pop up, and they're going to stick to the surface. Maybe they do this with their capsule, maybe they have some fimbriae, whatever it is, they're stuck to the surface and they form the capsule here in green. As the cells divide, they'll begin to form a dense mat of sticky extracellular matrix glycocalyx to the point at which their capsules will kind of start to become continuous with each other. Additional microbes will be attracted to this environment and this developing film, which will create a community of complex, various different organisms. You can see there's yellow ones and brown ones and red ones and all different shaped ones all together. And it's much more difficult to sterilize an area that has a biofilm to get the chemicals in to kill the bacteria than it is if it's just a free little individual bacteria. The cell envelope, the cell walls, and the cell membranes are the boundaries of the cell. And the boundary is marked by multiple layers. All together, all the cell wall, the cell membrane, all of those are known as the cell envelope. So big umbrella, cell envelope, and then underneath it, we have specific layers of the cell wall and cell membranes. The cell envelope is comprised of cell wall, cytoplasmic membrane, and any other outer layer, outer membrane that the bacteria provides. Now, there are two main types of cell envelope structures, and we alluded to this in our last chapter when we were talking about staining. The two main types are a gram-positive cell envelope and a gram-negative cell envelope. So let's go through this a little bit. The first thing that I notice on this picture is the cytoplasmic membrane shown here in yellow. And you'll notice that both gram-positive and gram-negative have this bottom yellow layer. And this is the cytoplasmic membrane that you might be thinking of from Bio 1 that has the phospholipids with the polar head and the nonpolar tail and maybe some proteins in blue mixed in. Both of them have that. As we move up, you'll see that there's a peptidoglycan layer, but it looks a little different in each. So you'll see on the left, the gram-positive purple one is very thick, whereas the one on the right, it's still there, but it's much thinner. Then finally, we'll notice that there is an outer membrane layer on the gram-negative cell envelope. Looks very similar to the cytoplasmic membrane. See, same idea of these phospholipids here, but there's no outer membrane on the gram-positive cell envelope. So big takeaways from this picture that I get is the gram-positive has 2 layers overall. Gram-negative will have 3. Both have the cytoplasmic membrane. Both have a peptidoglycan layer, though it's much thicker in the gram-positive than the gram-negative. And the gram-negative has an outer cell membrane layer. The cell wall is critical for determining the shape, keeping it a bacillus or a coccus. It provides structural support and it keeps the cell from bursting. It will regulate the osmotic pressure of how much water is coming in and going out. The cell wall is composed primarily of peptidoglycan. So that was that purple or pink layer we saw in the last picture. And it's critical that we determine whether there is a peptidoglycan layer, how thick it is, because many of our antibiotics will target the peptidoglycan layer. Without a functional cell wall, so if we do target it, that often leads to lysis or cell rupture of the cell. So if we look over here a little more on the chemistry of what peptidoglycan is, you can see that it is a series of glycan chains or multiple sugars here cross-linked together with peptides, and peptides are short proteins. So overall, this is a mixture of sugar and protein all together into one set of molecules. And if we zoom in further, you can see we have our sugar molecule out here. We have our short peptide here with some of these are amino acids you should have seen in our previous few chapters of alanine and lysine and glycine, forming a bridge to the next sugar molecule. A couple slides back when we were looking at the 2 different cell envelopes, you should have noticed that one was purple and one was pink. And that was alluding to the Gram stain. So this is a differential staining method that we discussed in the last chapter. And here are the steps that you would walk through in the lab to perform this particular stain. So before we even did the stain, the first thing we would need to do is fix the sample to the slide. So we would add the bacteria to a glass slide and we would either let it air dry or heat fix it. But either way, we need it to dry stuck onto the slide. Once we've made that smear, we will add crystal violet, filling the whole circle. If you think back to the last video at the end of chapter 2, filling the whole circle with crystal violet. Then we would wash that stain off, wash it with water, and move on to the next step. Then we would add iodine to this, and this is a mordant. So if you ever see using a mordant in a stain, it's probably going to be iodine. And this helps stabilize the dye in the peptidoglycan layer. It traps it in and forms a complex with the dye. Then we'll wash it off again, and then we add alcohol to it. And this is the decolorizer that is going to wash out any of the stain that's not stuck on. So if you look over here, gram-positive, we got the purple dye in and we trapped it in. We wash it out with alcohol, it's still purple. But since the gram-negative has a thinner cell wall, thinner peptidoglycan layer, there's not as much room for the dye to get stuck, not as much for the iodine to help it get stuck in. So then when we add the alcohol, all the purple washes out. At this point, if you had a gram-negative cell, it would appear clear, whereas a gram-positive would appear purple. Finally, we add safranin, a red or kind of pinkish dye, and this is known as the counter stain. It will come in. We don't know whether we have gram negative or gram positive, so we're going to stain everything. And if the bacteria is gram positive, adding the pink is not going to show up, because if we think back to elementary school, the purple is going to be darker and it's going to outweigh the pink. Okay. But if we had a gram negative cell and it was clear, adding pink will help us visualize it, improve the contrast when we look on the microscope. This stain is critical for determining the key components within the pathogen, and especially for determining what sort of antibiotics might be important to try. The gram-positive cell wall has that thick layer of peptidoglycan. It also contains teichoic acid and lipoteichoic acid, some extra molecules stuck in through the peptidoglycan. These molecules help the cell wall maintain its charge and keep its structure. Now, up to this point, we've discussed two main types of cell walls. We've had the gram-negative and the gram-positive, very binary. Now, that said, there are some non-typical cell walls. The main one being the mycobacterium. Mycobacteria contain peptidoglycan and therefore will stain purple, but they aren't truly gram-positive. They have their own unique lipids, mycolic acids, in them that are very waxy, and this makes them difficult to stain. So if you had a mycobacterium, they might come out vaguely purple, but they don't look quite right like a gram-positive stain typically would. If you suspect that you have a mycobacterium, you would perform an acid-fast stain. This is another differential stain that will use more intense molecules, more acidic molecules to force the stain in to see if it is indeed a mycobacterium. This is very important if you suspect your patient has leprosy or tuberculosis. Mycobacterium leprae and Mycobacterium tuberculosis are both, as they suggest, mycobacterium, and they're very difficult to treat if you have not determined their source. Now, mycoplasma are also bacteria that have unique or non-existent, I guess, cell walls. So for these types of bacteria, they still need cell structure, and to deal with this, they just make an extra strong plasma membrane. Their plasma membranes are fortified with sterols, or kind of cholesterol-like molecules, to make it more rigid, more resistant to lysis. One medically relevant mycoplasma is Mycoplasma pneumoniae. As the name suggests, it causes pneumonia or like lung infections. Additionally, I would note that there are some bacteria known as L forms that ordinarily will have a cell wall, but they can lose it as part of their life cycle and get it back, which, sounds a little counterintuitive, but it is very sneaky. If maybe we give the patient an antibiotic that targets the cell wall, these L-form bacteria could go, I don't need that cell wall, could lose it and survive. So it's very important that we can identify if we have an L-form bacteria. Gram-negative bacteria also have lipopolysaccharide, or LPS, in their outer membrane, shown out here. And LPS is known as an endotoxin because it causes your patient to have fever, often go into shock. So infections like meningitis or typhoid are very dangerous, partly because of the bacteria, but more so because of the endotoxins produced by the gram-negative LPS. The cytoplasmic membrane that all bacteria should have is comprised of phospholipids and proteins. And I found this interesting. Often in the little cartoons, you see that there's a ton of phospholipids and like 3 proteins. And in actuality, it's kind of flipped. More 30 to 40% phospholipids, 60 to 70% proteins. The cytoplasmic membrane is where the major energy reactions occur. So we haven't got to that chapter yet, but you can file it away when we talk about like the electron transport chain. Bacteria don't have a mitochondria. So where are they going to do the electron transport chain? We need a membrane to do that. And so they just use their cytoplasmic membrane as a dual usage. It provides like structure and the outside part of the cell. Oh, and it can also help with that energy reaction as well. It regulates transport because it's semi-permeable. It lets some stuff in and keeps other stuff out. It can help regulate how much water, how much salt, what comes in, what goes out. All right, so with that, it takes us through the first half of chapter 3. I'll see you back here next time.